Welcome to Medical Lab Science Quick Review. We present cerebrospinal fluid analysis for the clinical lab, followed by a Q&A quiz that follows our ACP study guide. Cerebrospinal fluid serves many important physiological functions, supplies nutrients to nervous tissue, removes metabolic waste products, maintains intracranial pressure, finally serves as a mechanical cushion for our central nervous system. For your information only, choroid plexuses of the two lateral ventricles and third and fourth ventricles of the brain produce CSF at the rate of 20 mils per hour. CSF flows through the subarachnoid space between the arachnoid and pia mater. The meninges of the central nervous system include the dura mater, which in Latin means hard mother. This tough covering protects the brain from the skull with both periosteal and meningeal dura mater coverings. The spinal cord is also protected from the vertebral canal with the dura mater. The arachnoid is a filamentous membrane, which means spider like in Latin. Pia mater means gentle mother, which gently covers the brain and spinal cord. CSF flows through the subarachnoid space between the pia mater and the arachnoid membrane. Tight-fitting endothelial cells of the choroid plexus forms an important structure called the blood-brain barrier, which protects the brain and spinal cord from harmful chemicals and substances in the blood that can harm nerve tissue. Unfortunately, the blood brain barrier also prevents antibodies and medications from entering the CSF. When the integrity of the blood brain barrier is compromised, as in meningitis and multiple sclerosis, harmful chemicals, leukocytes, and proteins enter the CSF and damage nerve tissue. Using sterile technique with a special kit, healthcare providers collect CSF by lumbar puncture between the third and fourth or fourth and fifth lumbar vertebrae. Healthcare providers collect CSF in CSF containers 1 through 4. CSF total volume is recorded for the hematology department. Tube 3 or 4 is reserved for the cell count because it is least likely to contain red blood cells from a traumatic tap. Sometimes the healthcare provider wants to assess their lumbar puncture technique by comparing red blood cell counts in containers 1 and 3. Expect it to be equal or higher in container number 1. Observe and record the color and appearance of the cerebrospinal fluid. Xanthochromia usually indicates the presence of red blood cell degradation products from a bleed into the central nervous system, and we later see a tinge of orange bilirubin in the CSF. Blood in the CSF may be from a traumatic tap from a vein. In this case, container number 1 is slightly bloody, hazy, or hemolyzed, and container 3 and 4 will be clear. As mentioned in our ACP study guide, if all of the CSF collection containers 1 through 4 appear light, pink, and hazy, then it is likely that the patient has a subarachnoid hemorrhage. Turbidity is a term used when cloudy body fluids obstruct light and we cannot visualize print behind the body fluid. Why is it usually not practical to use automated instruments to perform CSF cell counts? If you answered, because CSF cell counts are usually below the lower limits of linearity, you are correct. Background counts interfere with accurate cell counts in the lower range. For that reason, most CSF cell counts are performed using manual counting methods. In the past, we used the Neubauer cell counting chamber for CSF and body fluids. Note the 9 large squares with grids and small squares. The Neubauer cell counting equation is shown where we multiply the number of cells counted by a dilution factor if dilution is necessary, as it is for cloudy turbid specimens. Then divide this by the volume, which is measured in microliters. Remember the basic volume equation where the square area times the depth equals the volume? We count several squares that are 1 by 1 millimeter on each side, making it a square millimeter, and multiply it by the depth of zero point 0.1 millimeter to arrive at the volume of cubic millimeters, which equals the number of microliters or lambda that we use while counting. Introducing a modern innovation, the Kova Glastic slides. Let's take a close-up view of the grid. A clean and efficient technique is the glass stick plastic chamber with counting grids. Note the 9 by 9 grid. Nucleated white blood cells can be easily visualized at high dry magnification, ready for counting from right to left. Red blood cells are anucleated, making it distinct from the leukocytes. Lab protocol requires body fluid controls to be performed at least once a day if there is a body fluid cell count. These controls help you train your eye to distinguish the two types of cells. We count two leukocytes in square 1. We count two leukocytes in square 3. If a cell is on the border, count it if it falls on the right and bottom borders only to avoid overcounting. In square 4, we count two leukocytes on the right and bottom borders, but not on the top and left borders. 9 leukocytes were counted for 9 squares. The glass stick counting method involves a count times dilution factor times 90 divided by the number of squares counted. When we plug in 9 white blood cells counted for the 9 squares, we arrive at 810 divided by 9, which equals 90 leukocytes per microliter. In our example, 90 leukocytes per microliter is definitely higher than the ACP adult reference range, which is 0 to 5 per microliter. When performing duplicate cell counts, they should agree within 15 to 20% of each other, depending upon lab protocol. And if they don't agree, we mix the specimen well and then recount. And controls should rerun each day when there is a cell count. The cytospin centrifuge is a wonderful, indispensable lab instrument to efficiently prepare beautiful microscopic slides with a nice monolayer of cells, making it easy to stain and identify in the microscope. A special metal clip possesses 2 pins that allow a partial rotation in the cytocentrifuge. We can use a regular Fisher glass slide or a special black slide with a clear circle. A white plastic cytofunnel has a white filter paper backing with a hole for the CSF to pass through and attach to the circular area of the glass slide. The filter paper absorbs the excess cerebrospinal fluid. After assembling the parts, we clip it together. Using a hood or shield, we carefully dispense 3 drops of CSF using a sterile pipette. Use CSF container 3 or 4 that is designated for the cell count and differential. A cap will keep the CSF from spilling out. 1 drop of 22% refrigerated albumin may be used as a lubricant to keep fragile cells from disintegrating. Do not use albumin that has been kept out at room temp because it may act as a growth medium for contaminating bacteria. A cytocentrifuge, also known as cytospin, is a specialized centrifuge used to concentrate cells and body fluid specimens onto a microscope slide. Centrifugation gently pulls the CSF down the funnel and onto the slide, forming a concentrated monolayer of cells within the circle. After drying the slide on a 37 degrees centigrade hot plate, it is now ready for right Giemsa sustaining in the Hematech slide stainer. It is your option to use the Sysmex slide stainer if you use Sysmex glass slides. CSF in an adult patient contains 0 to 5 white blood cells per microliter, primarily lymphocytes and monocytes. Please follow along in this video with your Google search of images of CSF microscopy. In bacterial meningitis, the predominance of leukocytes are usually caused by neutrophils, so we see a neutrophilic pleocytosis. In viral and fungal meningitis, the predominance of leukocytes are usually lymphocytes and monocytes. Macrophages appear for phagocytosis and removal of cellular debris. In bacterial meningitis, we may see intra and extracellular bacteria. We need to correlate these observations with the CSF gram stain. In parasitic and fungal meningitis, we may see eosinophilia in the CSF as well as in the blood smear. Signs of a previous central nervous system hemorrhage may include xanthochromia in CSF seen in clear glass tube and pycnotic red blood cells seen under the microscope. CSF protein is commonly elevated in meningitis due to inflammation of the blood brain barrier. Central nervous system hemorrhages will also show an increased protein due to the presence of hemoglobin. Neurologic disorders such as multiple sclerosis may manifest elevated CSF protein results. Normally, CSF glucose is decreased by one-third of the serum glucose. However, CSF glucose is more markedly depressed in meningitis due to metabolism by bacteria or fungi and alterations of the glucose transport through a damaged blood-brain barrier. A very low CSF glucose, accompanied with a high CSF white count with a preponderance of neutrophils, strongly suggests bacterial meningitis, especially when supported by gram stain and culture results. In contrast, a normal CSF glucose value combined with an elevated CSF WBC count due to primary lymphocytes strongly suggests that the diagnosis is viral meningitis. CSF lactic acid is measured 10 to 24 milligrams per deciliter, and when it is above 25 milligrams per deciliter in bacterial, tubercular, and fungal meningitis, this is because of anaerobic metabolism of CSF to lactic acid when there is hypoxia. CSF lactate may be useful to monitor severe head injuries because of the high lactate in red blood cells. In these cases, may appear to be xanthochromic with small pycnotic red blood cells seen in microscopy. The ECP study guide had two questions regarding CSF lactate levels that are mildly elevated above 25 milligrams per deciliter and also markedly elevated above 35. As you might suspect, bacteria utilize glucose rapidly to lactate and markedly disrupt glucose transport through the blood-brain barrier. Note that this is not elevated in viral meningitis because viruses do not metabolize glucose. A brief note about elevated CSF proteins that are not caused by meningitis or CNS hemorrhage, but these are suspected to be caused by neurologic disorders. These cases may need further workup with CSF protein electrophoresis to assess for neurological disorders as shown in the chart. And one of the key words is oligoclonal bands. CSF myelin basic protein is valuable to demonstrate demyelination that is seen in these neurological disorders. We'll try to cover the India Ink exam and discuss Cryptococcus neoformans in a future video. Thank you for your time, attention, and support, and we hope to see you in the next video.